Hello, my name is Shraya. Together with my co-authors, Hong, Ryan, and Khadeeja, we are honored to present our research titled Comparative Experimental Validation of Human Emotion Recognition and Classification Using Physiological Signals. This work is intended for the Springer EHB 2025 proceedings.
Emotions fundamentally shape human behavior and decision making. While facial expressions can be controlled, physiological signals like EEG brainwaves, ECG heart rate, and EDA sweat glands provide objective, continuous indicators of emotional states. Our key challenge: achieving reproducibility and cross data consistency in emotion recognition research. This work matters for effective computing, mental health assessment, and human computer interaction. We focus on solving the cross dataset reproducibility challenge while effectively integrating multimodal physiological signals. So, we had four main objectives. First, evaluate both classical models like support vector machine and random forest alongside deep models such as convolutional neural networks and long short-term memory. Second, compare the performance across three distinct datasets: Seed, Dreamer, and our newly collected in-lab dataset. Third, identify how model architectures align with different signal characteristics. And finally, we develop a reproducible framework for multimodal emotion recognition.
To validate our models, we used three distinct datasets. First is the seed dataset, a large public benchmark with over 11,000 samples. It provides EEG-only data for a three-class emotion problem: negative, neutral, or positive. Second is the Dreamer dataset. It's much smaller with only 216 samples and is used for a two-class problem: low versus high valence. Finally, we collected our own in-lab dataset. This is a multimodal dataset using ECG, PPG, and EDA signals to classify sad versus happy emotions. For our in-lab dataset, we used a BioPack MP160 system along with a SenStream ring for our multimodal signal acquisition. The experimental protocol for our 6 participants were consistent. Each session started with a baseline period, followed by video clips designed to elicit happy and sad emotions. Furthermore, we used a rotating Latin square system. This ensures that the previous video clip doesn't influence the next. The specific signals we recorded were ECG, PPG, and EDA. These were all synchronized and sampled at 256 Hz in a controlled environment to ensure high-quality, aligned data for our analysis. Our methodology followed two main phases. First, we applied standardized preprocessing across all three datasets. This was crucial for ensuring consistent data quality and involved filtering, segmentation, and z-score normalization. Second was feature extraction, which we had to tailor for each dataset. For seed, we used the provided differential entropy features, which was extracted from 4-second windows across 5 EEG bands and then averaged per trial. For Dreamer, we used a branched approach. For classical models, we used simple per-channel statistical features. For deep models, we fed them the full temporal EEG sequences. We also applied SMOTE to handle the dataset's class imbalance. And for our in-lab dataset, we segmented the data into 10-second windows and transformed the raw signals into meaningful physiological features like HRV, spectral, and conductance metrics, which were then normalized against each participant's baseline. And here's a more detailed breakdown of those processing steps. First, artifact removal and filtering. For the EEG data in Seed and Dreamer, this meant a bandpass filter from 1 to 50 Hz to remove drift, muscle, and line noise. For our in-lab signals, we filtered out motion artifacts and baseline drift. Second, synchronization and resampling. This was key for our in-lab data. We synchronized the signals from the BioPack and SenStream devices and resampled them all to a uniform 256 Hz. Third, normalization and baseline correction. We applied z-score normalization within each subject and also used baseline correction relative to the resting period to reduce variability between participants. And finally, data balancing. This was specific to the small Dreamer dataset, where we applied SMOTE during training to mitigate its valence class imbalance. This complete methodology ensured our data was clean, consistent, and ready for modeling.
Moving on to models and evaluation, we used 4 models: SVM, random forest, CNN, and LSTM. SVM captured nonlinear patterns while random forest improved robustness through ensemble averaging. CNN learned spatial EEG features, and LSTM modeled short temporal dynamics. Seed and Dreamer used a 70-30 split, and the in-lab dataset used a fivefold cross-validation, evaluated on accuracy, precision, recall, F1, and RSC AUC scores. This is our signal processing workflow diagram, which represents our complete signal processing and feature extraction pipeline for all the datasets. The workflow begins with raw physiological signals EEG, ECG, PPG, and EDA, which undergo artifact removal and bandpass filtering to eliminate drift, noise, and muscle interference. Next, all signals were synchronized and resampled to a uniform late for temporal alignment. They were then segmented into fixed windows, which is 4 seconds for seed and dreamer and 10 seconds for the in-lab data. Normalization and baseline correction were applied within each subject to minimize interperson variability. Finally, feature extraction converts the signals into usable representations, like differential entropy for seed, and normalized EEG sequences for dreamer, and HRV spectral and conductance features for the in-lab dataset. Moving on to results for seed, CNN achieved the highest accuracy at 90.2%, while LSTM was lowest at 60.24%. CNN captured spatial EEG patterns effectively, which align with valence-related brain activity. LSTM underperformed because seed's short per second segments lacked sufficient temporal continuity for sequence learning. Moving on to Dreamer, Random Forest led with 83.8%, while CNN had the lowest accuracy at 61.1%. RF's ensemble structure handled nonlinear and low-dimensional EEG features effectively. CNN struggled because Dreamer's signals lacked strong spatial organization, limiting convolutional feature learning. Finally, for in-lab data sets, Random Forest achieved the highest accuracy at 98.4%, while LSTM was the lowest at 66.94%. RF captured complex relationships among ECG, PPG, and EDA, features with strong separability between happy states and sad states. LSTM performed poorly since physiological segments were short and mostly steady state, offering very little temporal variation.
Let's dive into the metrics a little deeper. On seed, CNN showed the lowest variance with higher consistent performance, effectively capturing spatial structured EEG features and achieving 90.2 accuracy. On Dreamer, Random Forest achieved strong generalization despite fewer samples at 83.8 accuracy, while LSTM balanced precision and recall but lacked long-term context. Lastly, our in-lab data set, Random Forest delivered a 98.4% accuracy with minimal error. The multimodal fusion of ECG, PPG, and EDA significantly enhanced robustness under these controlled conditions. On seed, CNN achieved 90.2 accuracy with the area under the curve at 0.98. The confusion matrix shows tight diagonal clustering, demonstrating excellent spatial pattern recognition in EEG data. For the Dreamer dataset, Random Forest led a 83.8% with a 0.93 area under the curve, showing superior generalization on smaller datasets. The confusion matrix clearly shows valence separation. Our in-lab multimodal fusion delivered 98.4 accuracy with Random Forest and a near perfection of 0.9987 area under the curve. The confusion matrix shows exceptional emotion separation. Multimodal integration clearly works.
Moving on to the next section, our study showed that model performance depended more on the type of signals than on the model complexity. CNN worked best for the EEG data because it captures special brain patterns linked to the emotions. Moreover, the random forest performed strongly on multimodal signals, showing great consistency and generalization, whereas the LSTM didn't do as well since our data segments were short and limiting temporal learning. So overall, standardized preprocessing helped reduce subject differences and improves reliability. So our datasets were small and recorded in controlled lab settings, so results may not fully generalize to larger populations. Average features in SEED and the Dreamer dataset limited temporal analysis affecting LSTM results. So we also focused only on EEG and physiological signals without any facial or voice cues. Cross-subject and real-world validations are still needed in the future work. In these future slides, we plan to expand to larger and more diverse datasets for better generalization. We will integrate multimodal fusion combining the three modalities, which are EEG, ECG, and EDA, and behavioral cues like facial and voice signals. We also aim to test newer models like transformers to capture emotional changes more effectively. Finally, we will develop lightweight, real-time systems for wearable devices. To conclude, we built a unified framework for emotion recognition using EEG and physiological signals. CNN achieved the best results for spatial EEG data, while Random Forest was most reliable for multimodal signals. Our results show that aligning model design with the signal type is key for accuracy. This framework is a strong step towards robust real-time emotion recognition. Thank you for watching this presentation. I appreciate your time and interest in our work, and I hope it contributes to ongoing research.